Hi, and welcome to the Disease-Relevant Translation Models in Ocular Drug Development Types, Challenges, Advantages, and Alternatives webinar. This is our second in a series of 4 webinars hosted by FFB, Foundation Fighting Blindness, and Odelia Therapeutics. My name is Ashley Winslow. I'm CSO of Odelia Therapeutics. If you're not familiar with our organization, I invite you to visit our website and sign up maybe for our new newsletter, which we just launched this week. We're a nonprofit biotech focused on accelerating therapeutic development for rare disease, regardless of number of people with the disease or the opportunity for a company to make money. So we think about things a little bit differently. Please feel free to connect with us, start a conversation, or share your experiences in the rare disease space. So before I turn things over to our co-host, Dr. Chad Jackson, who is the Director of Preclinical Translational Research at Foundation Fighting Blindness and our moderator for the session, I'd like to review some logistical details for this session. Currently, all participant lines are in listen-only mode. To ask questions during this session, you can use the Q&A feature on the Zoom control bar to type your question. That's at the bottom right, and it is different from the chat function. So make sure you find the right button if you want to ask a question. This will go a long way in helping us organize the discussion today. And today's session is being recorded and will be available for future viewing at a later time on Foundation Fighting Blindness, their website, as well as Odelia's website. We currently have webinar number 1 up on YouTube now. So please join us, and we look forward to webinar number 2. With that, I'd like to introduce Dr. Jackson. It's your floor. Go ahead.
Thank you so much, Dr. Winslow, for the introduction, and greetings to everyone. Thank you for joining our webinar series. Welcome to the Disease-Relevant Translational Models in Ocular Drug Development Types, Challenges, Advantages, and Alternatives webinar. The Foundation is proud to partner with Odelia Therapeutics to bring you this webinar series. The purpose of this series is to share tangible preclinical and translational research and development information to our drug discovery community, focused on providing solutions to inherited retinal disease, as well as the broader rare disease field. Last month, we hosted our first webinar series entitled Tools for Preclinical Development, Creating a Target Product Profile and Effectively Engaging Regulatory Entities. As Dr. Winslow mentioned, this video can be found on the Foundation Fighting Blindness' YouTube page for your viewing. Today, we will discuss the advantages and disadvantages of certain retinal disease models and their utility as tools to move new therapies toward investigational new drug filings and clinical use. Whether you are developing a small molecule, a cell therapy, or genetic technology, these assets will require significant assay and disease models for testing before human use will be considered. We are thankful for the doctors Kylan Dula, Nina Hayter, and Gus Aguirre, who will lead us through today's meeting, which covers retinal organoids to small and large animal models. Throughout today's event, we will have some polling questions for the audience. Your participation in this is anonymous, of course, and we find this as a great way for us to understand who is joining the event and get some granular information as we step through the topics. We kindly ask that you participate by answering the questions when they are presented. In order to kick this off, let's start with the polling question now. So the polling question that's about to be presented is, what is your role in drug development? Leila, could you put up the poll for the people? And we'll just give you all a moment just to actually answer these questions from the poll before we introduce our first speaker. This is very interesting to see the poll going back and forth to see who is actually on the call. And again, we are very happy that you joined the event because we feel like this engagement goes so far in helping our community actually see progress and get therapies and treatments to those who need it most. All right, according to our poll, most people on the call are academic-based researchers, followed by biotech, followed by patient groups and investment groups, and others. And we welcome you all to this event. Now it's time for our first speaker. So we have Dr. Nina Hayter. She is an associate professor at Harvard University. Dr. Hayter is a visionary and creative leader bringing medical science and healthcare and developmental breakthroughs for therapies that have global impact. She has received a PhD from the University of Iowa in genetics. She was a part of the Human Genome Project and was involved in identifying over a dozen human disease-associated genes. Dr. Hayter directs a cutting-edge genetics and gene therapy laboratory at Harvard Medical School, developing and identifying viable therapeutic options for blindness, such as retinitis pigmentosa, age-related macular degeneration, and diabetic retinopathy. Her team is the first to develop a novel broad-spectrum gene therapy to treat multiple forms of blindness and has a U.S. patent issued and 2 FDA approvals for orphan disease designations in gene therapy. Dr. Hayter's talk is entitled Modeling Human Disease and Developing Effective Therapies for Human Retinal Disease. Dr. Hayter, thank you for joining us today. The floor is yours.
Thank you very much, Chad, for the invitation, and I'm thankful to have this opportunity to talk to you about our work and about the steps that we took and preclinical work that's important and essential in order to develop effective and viable therapies for treating retinal diseases. I'm going to. So I want to start to just give you an overview of what we're going to talk about today. Focuses on retinal diseases, but I want to give you an overview of the current state of gene therapies, what technologies are available, pros and cons of different model systems in preclinical studies, and end with what considerations are important for preclinical studies. So most of you probably know the demographics of this slide very well, but I'd like to start with this tidbit, and hopefully you can see my cursor as your pointer here too. So the DNA between any 2 individuals is pretty identical. All of us sitting here, and when I started with the Human Genome Project during my PhD, and as we were discovering this phenomenon, it was very fascinating to me that only 0.1% of our DNA differs, and those single nucleotide polymorphisms give us all the variation we see in humans that are just normal and also contribute towards disease. 60% of blindness in infants is caused by some kind of inherited eye disease. And these can be cataracts, congenital glaucoma, optic atrophy, and various eye malformations, and including RP, which is retinitis pigmentosa. And that's a disease that I've been studying in my lab for many years now. In adults, glaucoma and AMD are 2 of the leading causes of blindness and severe vision loss in our societies. Common vision problems can also occur, such as strabismus, amblyopia, myopia, hyperopia, and astigmatism. Now, when thinking about model systems, there are over 20 different models for RP that have been developed and over 30 different models for AMD, trying to capture the disease phenotype and recapitulate those diseases. This is a graph from Retinal that I like to refer back to and look at periodically and see. This was updated in 2019. I think we're kind of coming to a halt here, where we've maximized the number of cloned retinal diseases that were both identified and mapped. That said, with the genome sequenced and the number of patients that we still don't have an idea of which retinal diseases are causing their phenotype, there's still work to be done in this area. However, a focus has shifted in the last decade or so in really characterizing the models and understanding the disease pathology. I'm going to give you a few very general, high-level slides to just show you an understanding of what goes into developing gene therapy studies and the current state of therapeutics in the world. The preclinical research that many of us are interested in, invested in, and can span our entire careers doing is to understand the disease mechanism, targeting identification, doing these animal studies and tox screens. And that can take 20 to 40 years. Phase 1 is a safety in humans, requires about 10 patients, and then phase 2 is a small scale efficacy often, and that is in the teens, 10s to 20s of patients. And then phase 3 is the larger scale efficacy that's in the hundreds of patients. And the cost for doing these studies, as well as the time frame, is quite expensive and can take 5 to 6 years if speed lined, right? with a very low success rate right now, 10% success rate. So part of our preclinical work that we invest a lot of time in is going to be to how can we ensure a greater success rate here. That's one of the key features that we think about in my laboratory is how can we develop something that's viable, effective, and will increase our success rate of what we can take to the clinic. In the US, FDA is our gatekeeper, and there's review at every single step. And we've been fortunate to start that process with the work that I've been doing. And those guidelines, I think, are essential and important in helping us develop the viable therapeutics. There are many ways that gene therapies are now being delivered, and I want to go over these different vehicles of delivery before we move on to what kind of trials are ongoing currently. So the first one is adenovirus, and these are viral versus non-viral, so we'll start with the viral. So adenovirus is about a 35 kb double-stranded DNA. It has a very mild respiratory tract infection associated with it, and it does not integrate into the host genome, which is an advantage, and it can infect dividing and non-dividing cells. That's also an advantage of using adenovirus. The insert size is a limitation. It's only up to 2 kb. However, if you have a small gene that you're working with, this is something that's actually quite commonly used, as we'll see in the next couple of slides. You can get a really high titer. There are up to 40 different serotypes, and number 2 and 5 seem to be the most common. Then we switch to adeno-associated virus, or AAV. This is also non-pathogenic. It's non-enveloped. It's a single strand of DNA. So it's the parvovirus assisted by an adenovirus. There are about 9 different serotypes, and the cargo size has increased a little bit more from adenovirus to 4.7 kb. It's still kind of small for a lot of genes though, but it has advantages, and it's actually becoming fast the more common vehicle of delivery. Retroviruses are enveloped RNA virus, except for the lentivirus, which I put down. They can only transform mitotic cells. Lentivirus can transform into non-dividing cells, which gives it an advantage and therefore has been used in therapeutics recently. The insert size is about 7.5 kb, which is bigger than for adeno or AAV. And these are all considerations when thinking about what gene of interest, what disease, what target. And another bonus feature for these retroviruses is they're self-inactivating components that can be put in. There are also non-viral vehicles of delivery like plasmid naked DNA that have been used to get into the cell, nanoparticles that can have a cargo size of up to 20 kb, so that's advantageous for large genes such as the USHERS2 in the eye field. And then there's shRNAs, which are important for gene silencing. Current clinical trials that have been going on have increased over the last couple of decades. So if we look at this chart from 2000 to 2018, we started about 96, and we're currently, this is end of 2018, over 200 different clinical trials happening in the gene therapy space. These are not all ocular. This is more of a general slide to show you. And I like to just mention this timeline here that in 1953 was the discovery of the double-stranded DNA. And it took a couple of decades to construct the retrovirus system, conceptualize gene therapy. And the first clinical trial, gene therapy trial, was in 1992. So early 1990s were the first gene therapy trials that came about. And since then, many issues had come up, and gene therapy kind of saw this halt in reassessing what do we need to do to make a safer therapeutic possible. And in the last few years, actually, we've seen a greater increase in these therapeutic trials for using gene therapy as a mechanism. And they seem to be so far safer than what was before. So that's exciting and encouraging. Current geographical distribution of gene therapy trials around the world, just so you have an idea, most of it, probably not surprising to this audience, is in the US, about 60% or more. And ocular, sorry, my mouse cursor moves faster than me. Ocular diseases are about 37 different trials. So most of them, as probably we're not too unfamiliar with, is it going to be in cancer therapeutics, and monogenic diseases take up the second largest chunk. And ocular in particular are a bit lower, but 37 different trials. Now, if we look at what different vectors or vehicles of delivery and what type of therapeutics are coming out, adenovirus is the most common, most meaning 18%. It has the highest number. It's not anywhere near like a 50%. So about 540 different trials using adenovirus. Retroviruses are the second largest type of vehicle of delivery being used, and naked plasmid DNA is actually higher up there than, I don't know if you thought this when I was doing my research that I realized. And AAV is fast becoming something that is interesting and talked about. And maybe at the end of when we look at questions, we can think about viral versus non-viral, because there's a stronger push, I would say, in the last couple of years to really pull something that's non-viral. And that's where plasmid or naked DNA are coming in as something, and nanoparticles are seeing a resurgence, if you will. If we think about what type of targets, the antigen targets are probably 17%, the highest number here, and then receptors are the second highest, and then cytokines as what are gene therapy targets. In the eye in particular, this is a table, and I've put on the top here for your reference, which the journal article that this table was taken from, which I think is a great summary table. It was just recently published in 2019, so it's pretty up to date, but it gives you an idea of what company, these are specifically for ocular gene therapeutics, the programs and the products, the disease mechanisms that are covered, the vector technology. And if you go down the list, you'll see AAV2 for a lot of this, and AAV7-8. So AAV is becoming very common. You also have some lentivirus vectors that are common. And then route of administration has been typically subretinal. And I know with the clinicians, they're very interested in having more intravitreal for a variety of very important reasons, and mainly related to the patient and their disease phenotype to begin with, and to have just less damage in that retina. This table also tells you that a lot of these are in between phase 1, phase 2 clinical trials, and this number is going to keep increasing, and I think that's encouraging that there are a lot of different options for therapies that are now reaching post preclinical into actually the clinical trial phase. I'm going to spend the next few slides going over different models and the pros and cons of using them for these preclinical genetic studies. And this first slide, which covers our species, this Homo sapiens, humans, Canine will be giving you a great talk after this, and I'm just going to very briefly talk about advantages. Of course, is directly relevant to human disease, and I think his focus will probably be the testing of therapeutic targets, which is very exciting to have these organoids in addition, use precision medicine-based technologies to harness what we can from human cells and use, and they're actually great advantage to using these human cells in some cases where you don't have a model available to test to. Disadvantages of the system is that their organoids are typically in a developmental stage, so not exactly ideal for the aging diseases. Adult iPS cells may not always be optimal, and lifespan's limited, and there's a lot of genetic heterogeneity. Primates are also used in limited capacity. The nice thing about primates is they have a fovea. That's oftentimes, I'm a mouser, and you'll see a little bit of my work that we've been doing, but it's the closest model to humans. It's often used in behavior studies, but in context with this presentation, it's a model that's become important to use for safety and toxicity if needed on a higher primate level. Disadvantage is not as developed as the mouse. It is costly. There are breeding issues and times of breedings, and there are no naturally known occurring mutants in them. The mini pig has become almost a favorite, I think, for toxicology studies. So the advantages of using the mini pig for preclinical studies is their physiology really resembles the humans better than the rodents, for example. They're used in therapies to look at pharmacokinetics and metabolism. They're similar eye size to humans, which is good, and they're used to evaluate toxicology of drugs or biologics. Disadvantages might be that they're not as well as developed a model as some of the rodent models. They do have immune issues, which can compound your readout and your effect. So that's important consideration to make. And they can be costly and a little more challenging to take care of. The dog, which you have the premier expert that will be speaking later, Gus Aguirre, on. I'm just, again, briefly pointing out the advantages for this and disadvantages. They are naturally occurring models in dog for eye diseases, and they're used for toxicology studies, but also for safety. So looking at safety in gene therapies, it's been a tremendous model that has both because they have naturally occurring mutants that are similar to humans, has been, a great tool for using in preclinical work. They're expensive to breed and maintain, and the lifespan limitations might prove to be some disadvantages depending on the study you're trying to do. Mouse is a very common go-to model for preclinical work on gene therapies and in retinal diseases or eye diseases in general. There have been really probably more developed than any other model in many ways because there are numerous transgenics, there are reporter genes. You can knock a gene out, you can knock the gene in. There are many mutagenesis screens that were performed that then have a plethora of animals to work with. And so they give you really a lot of different models for ocular diseases. And disadvantages when using the mouse is the organ size compared to humans, and the physiology just does not resemble as close to humans as other organisms. I'd like to put this strain up to remind any of you that are working with mice or will work with mice. There are over 100 different inbred strains of mice. When we do gene therapy studies in mice, one of the most critical and important facts to know is that there are genetic differences between these strains, and this is normal difference across the strains. And some strains actually do harbor specific mutations that in the last few years have come out, and we've become more aware. Even the C57 Black 6, whether it's a Jackson Labs or, for example, a Charles River, might harbor an RD8 mutation, which is retinal degeneration. So it's important to know the genetic background of the animals you're studying and what their quote unquote normal looks like. And because of this genetic diversity, we were actually able to perform our modifier gene studies, which I'm just going to give you a couple slide information on that. Jackson Labs has a phenomenal webpage if you need to look anything up to do with a mouse in terms of phenotype, genotype, expression. There's a lot of QTL data archived there. A lot of us, if we have mice we don't work with, we'll work with Jackson Labs to store the mice or cryopreserve them there. So this is a tremendous resource to the scientific community, and in for all diseases, but in particular for ocular diseases. Mouse tend to recapitulate in general the phenotype pretty well of what a human retinal disease looks like. So it's an advantage for that using as a model. The rat, which is similar to the mouse, but bigger, and their physiology actually represents human physiology better than a mouse. So for some physiological studies, this may be a better model. For example, people that study diabetic retinopathy and they want to look at the diabetic end of this and the impact, this is better. It's also used in toxicology studies. Its genome and its number of different strains are not as developed as mice, and so therefore it's not as common as the mouse to use. But there is a rat genome database that is also really well annotated and gives a tremendous amount of information in terms of the genes and the strains of rat and the different diseases available to study. The rabbit is another larger rodent that is used for safety toxicology studies. It has a larger eye than the other rodents, so it's closer in size to the human without jumping into the mini pig. It's good for physiological studies and pharmacology as well as tox studies. Disadvantages are there are no really genetic models and they cost more than the rodents. So you want to be really careful in terms of thinking why you want to use this rabbit model. So it's great if you want to do just the tox study or to look at the effect of a drug. And now I'm going to shift gears slightly and focus more in on some of the research that we've done and in looking at gene therapeutics. And in my lab, we've studied modifier genes and their impact on disease. And it started with this major shift in how we thought about monogenic diseases. And there are very few monogenic diseases where it's a direct correlation of a mutation resulting 100% of the time into a particular phenotype. Instead, what we've learned over the last decade is that disease outcome is determined by mutational load. And that mutational load is the primary mutation as well as modifier alleles or other factors such as epigenetic factors that have an impact on an entire gene network. So if we look on the right panel of the three different types, typical types of therapeutics, we have gene augmentation, which is replacing a mutated gene with the normal version of the gene. And that is present in addition to the mutant gene. We have gene inhibition. This is their CRISPR gene silencing where we're inhibiting the function of that gene. And the new gene will block the expression of the mutant gene. CRISPR has a lot of advantages and we can talk about that later. I didn't put a huge effort into that right now. It's great for dominant diseases. It's great for if you have a double negative, but it's still a newer technology that needs to be fleshed out. So there's still off-target effect concerns that can happen with that. Now, the modifier therapy is completely a new therapeutic type. So this is in the presence of the mutation. This is another gene. So it's not gene augmentation. It's not the same gene. We're not doing actually anything with the mutant gene. In the presence of the mutant gene, we're adding another master regulator. And what that master regulator does is impact an entire network of genes. Hundreds of genes are impacted here. And that's a real advantage here, where the modifier gene is something that can alter the phenotype. It can enhance or suppress it. It can function in a parallel or similar pathway, potentially can be far more potent than therapeutic than the original gene replacement. So in our approach, we start with the human disease gene, look at the defect, develop in the mouse a disease model and examine the pathology of the disease. And we've spent a tremendous effort looking at gene networks and gene pathways, realizing that that mutation is within the context of the rest of the genes that are expressed in the system, and using those different pieces of knowledge to develop targeted therapeutics. In our lab, we study prevention of disease, attenuation, guided gene cell replacement therapeutics, and the modifier therapeutics, all towards the goal of exploring and developing the best, most efficient therapeutics. So in retinitis pigmentosa, as I'm sure this audience knows well, it causes blindness in 1 to 1,000 or 1 to 3,000 individuals worldwide. And there are over 200 genes that can cause RP. So mutations in any one of these can cause the same disease. And that's where my lab started thinking about, let's look at the phenotype, that we have the same phenotype occurring regardless of these different 200 mutations. There are genes known that function in these pathways. And while there are really great clinical trials ongoing for current gene replacement therapies, they're only going to target a few of these hundreds of genes, right? And 40% of patients that have RP can't have a molecular diagnosis. They're novel mutations or spontaneous mutations. They can't get that molecular diagnosis, so they would have actually no option. That's what drove the research here. So what the genetic modifier therapy was able to do is alleviate the need to test or develop hundreds of different specific individual gene therapies. The modifier therapy is a powerful therapeutic as it can have an impact on multiple disease outcomes. So in the presence of the mutation, it can impact disease. The panel on the right here shows when we had an RD7 mouse, which has retinal spots, and we were able to administer the therapeutic and alleviate the spots. Histologically, they present with whorls and rosettes, and those also resolved. And then functionally by ERG, photopic and scotopic, we were able to improve visual function. And all of this happened by resetting multiple gene networks. So our therapeutic is a nuclear hormone receptor that regulates hundreds of gene networks, including those for oxidative stress, metabolism, phototransduction, so the function of the photoreceptor cells, cell survival, neuroprotection, etc. I wanted to show this slide to you to illustrate a couple of different specifics on readout assays that are disease specific. So we look at these two particular assays, we're looking at the fundus by indirect ophthalmoscopy, as well as immunohistochemistry on tissues to look at the expression of photoreceptor opsin genes. And sometimes we'll have a model here where there's no difference. In fact, in a few of these models where fundus is not the best readout because there's no change in the fundus because they didn't have a fundus phenotype to begin with. And other models where there is a fundus phenotype, we can look for resolution of that. And that's illustrated in a couple of these models. What was very clear for us at the molecular level is when we started looking either by RNA or in this case by protein and immunohistochemistry, as we could see in untreated where they did not express the photoreceptor genes. Now the cells were there where they were just not expressing the opsins, and they were able to re-express opsins following therapy. So when thinking about which model might be best for preclinical studies, there's several considerations that are important. How well do they recapitulate that human disease phenotype? Is the gene actually expressed in the species of choice that you want to use for a model? And what is the goal? Is the goal to look at the phenotype, efficacy, safety, pharmacology studies, or tox studies? Knowing those will help determine what would be the best models. Readout assays are really important. We work very closely with our clinical collaborators to see that the assays that we are assessing in our mouse models are actually translatable and would be something that could be assessed also in the humans. So while we can do much more in the mouse and look at a molecular level and look at tissue for we consider the patient and think, what other readouts can we do that would tell us and enlighten us at all on how that readout would look like in our model compared to the patient. Route of administration is something that we're also very cognizant of. Subretinal has been a go-to, and we've used that, but we also look at intravitreal injections or devices, slow release mechanisms of administering the therapeutic. And cost, of course, are very important when you consider your model. Generally, the larger the animal, the more the cost. But all of these together need to be considered in order to determine what would be the best model to use and how to best design those preclinical studies. So in conclusion, I think it's always an exciting time to do gene therapy work. We're in a period of time where as science occurs, it's what's hot today and a few years from now might be obsolete, and there'll be something newer and better. And it's great to be in that period of time where we are exploring the different vehicles of delivery for gene therapy. We've done tremendous work in terms of designing and developing models, and preclinical research is essential. That takes the bulk of the time for gene therapies to reach the clinic, is the preclinical work that lays the foundation to understand more about the disease, to look at safety, efficacy, whether it's of the therapeutic target or of the vehicle of gene delivery, and the different cell types that they might impact and how that might affect disease outcome. And managing and minimizing immune response is not a trivial matter and is something that's important to consider. So as research and technology grow and expand, interesting and exciting to be in this phase of really going towards precision medicine and personalized medicine for prognostics before treatment. I didn't touch a lot on that part, but we're also looking at organoids and how we can make precision medicine for developing better therapeutics in ocular diseases too. Thank you very much for your time. Happy to take questions.
Awesome. And Dr. Hare, thank you so much for the presentation. Really appreciate it. We have two questions here that have been queued up. We'll take these and then move on with the rest of the presentations. But the first one, I'm going to go in reverse order here for the audience. What are the safety risks for the gene modifier therapy as this approach impacts hundreds of genes?
That's a great question. We are exploring that still. We just received another round of funding to do these type of studies. So in our work thus far, we don't see any major issues in terms of toxicity. Of course, this is a mouse. We are doing tox studies actually using mini pig. And so in terms of this receptor, because it has a broad spectrum effect, one thing to remember, and we published this several years ago, there's auto feedback loops often. So while it regulates hundreds of these networks, what it's doing is resetting a homeostatic state of the retina. When you reset homeostasis, you no longer, it's actually taking advantage of all the natural networks in the genes because you're impacting a little dose, if you will, like a spark plug. That's how I look at it. You just spark it and it goes. And then when it needs to reset, it resets. It's not that the system will be pumped up for long periods of time. So I think that although on the onset, we thought this could have, catastrophic and just, global issues, it actually doesn't. And it actually, what we're learning, and we're continuing to do the studies, so I don't think the final word is out yet, but what's encouraging is that this resetting a homeostatic state of these cells, and these are cells that have the mutation versus, and also the adjacent cells that don't, that the entire retinal environment has become a healthier state.
All right, thank you for that. And this next question actually is was directed at the whole panel, and we'll just ask it now just to sort of meet the need of the audience. And that is, what is your opinion on biodistribution models for the retina? For example, non-human primate or pig, and specifically for topical drops for small molecules intended to reach the retina.
So the question is, what would be the best models for
I think so. So the way I sort of take it is that I think we see a lot of things of sort of the eye drop for therapy is sort of the brass ring that everybody's going for.
It is. Yeah.
Even though you showed a little bit of some information about subretinal injection and how physicians are sort of leaning toward, like, we really would rather do intravitreal injection. So I think at the end of the day, whether we're talking about neuroprotection or maybe in the future some type of gene therapy, if you could just put a droplet on and actually get to the site location of the retina, that would be the preferred thing. But as people are developing those types of technologies, what is the preferred model in doing something in the eye drop model?
So I don't think there is a one go-to preferred, and I'd let the other panelists chime in too, but in my opinion, we're exploring that actually with eye drops and contact lens delivery too, how to get it in without having to put a needle through. And so what you need to consider is the target is the diluent, the product itself, what's going in. That's almost as important, because you need to look at corneal thickness and you want it to reach all the way back to the retina. Then you have to think of viscosity. There's a lot of chemistry and biochemistry, kinetics that need to be considered when you think of the models. So each of the models that I talked about today have different distances. I tend to start with the mouse because it's convenient and small, and I've even made mouse contact lens before that are tiny, but just to test. But so I don't know that there is a preferred. No, I know a lot of people use rabbit because it's bigger than the mouse or any other rodent, and it's kind of closer. And you have to again go with cost effectiveness, what you're putting in. So I would say if there was one preferred right now for something like drops, it might be rabbit to start. And then you would go higher when you want to look at safety. But Gus, maybe you could comment on that.
I think a rat is an ideal model to initially test the principle that something put in the eye can get to the back. Once you move and you show that it does get there, you have to take into account eye size. And a rat eye is too small and the volumes are too little in terms of a drug getting through. I have concerns with it. I think the rabbit is perfect. The trouble with the rabbit is that its retinal circulation is very different. Mm-hmm. So that what might stick around in the vitreous more in a rabbit, other than a, let's say, a non-human primate or a dog or a cat, is a little different from what would be in a human. Now, non-human primates are a problem in that you are having to put drops on them. Handling issues are not easy. And this may be something that is limited. So you need something that is large enough, that has a human-sized eye, or comparable to it, and that it is manageable. And I think then, but I think the rat would be the ideal model, or a mouse to only test. If you put something in the eye, will it get to the back and have the therapeutic effect?
Okay, great. Thank you for that. Okay, in the interest of time, we'll conclude that session. Thank you again, Dr. Hager, for that. And actually, we have one polling question as a follow-up to the presentation. Liana, could you load that polling question, please? And this polling question is, with regards to mouse models for the diseases you most commonly work with, how often do they mirror human disease characteristics? We'll give it about another 20 seconds or so for people to answer, and then we will end the polling and move on to our next. It's very interesting. We have about 50-50% of the time, 50-50 leading, and rarely, if ever, coming in second. So here are the results from the poll. So we see that's about 50% of the time. You see that your models are reflecting the human disease characteristics. So it's very interesting. So our next speaker, we have Dr. Kalyan Dula, is a Senior Director of Ophthalmology, Non-Clinical Research at ProQR Therapeutics. He leads and manages multiple highly complex projects and programs encompassing the development of the ophthalmology franchise. And he really works in the whole sort of breadth of preclinical development all the way up into these types of therapies enter the clinic. He has a major focus on pharmacology and translation of preclinical science. Dr. Dula's talk is entitled Retinal Organoid Models for Drug Development. Callie, the floor is yours. Take it away. You're still on mute, I believe. There we go.
Thank you, Chad, for the nice introduction. Let me know when you see my screen, the right screen.
You can see it.
Yes, we can see your screen.
Hello, good afternoon, everyone. Chad already gave a nice introduction, so I'm going to skip and move to the topic. To start with, what are retinal organoids? Because that is what I'm going to talk about in this presentation. These are essentially lab-grown mini retinas made from stem cells or retinal-induced pluripotent stem cells. Here are a few images of typical organoids. They're approximately 1 to 2 millimeter in diameter. They contain most of the retinal cell types. You can see the photoreceptors, especially the outer segments, even under bright field microscope. These structures are often referred as brush borders. Here is a cross-section of an organoid where the nuclei were stained with DAPI. You can clearly see the inner nuclear layer, outer nuclear layer, and the ganglion cell layer. In the next image, they are stained for cone photoreceptor marker, red-green opsin. and then for rod cell marker rhodopsin. As you could see, these retinal organoids tend to contain more rods than cones. And then here, you see organoids stained for neural marker TUJ1. Also, as you have already noticed, the orientation of the cell layers inside these organoids is kind of reversed inside out. And the retinal pigment epithelium, RPE, is missing in these structures. That is because in organoids, RPE does not generate as a monolayer apical to the photoreceptors. Some organoids lack the RPE, as shown here altogether, or they do contain RPE, but in patches. I have borrowed some nice electron microscope images of these retinal organoids from Ovando Roche et al. This is the surface of the retinal organoid, displaying the inner and outer segments and the connecting cilium. In organoids, they do form synapses. Here is a cone photoreceptor stained by LM opsin in magnetar, and it's a ribbon synapse stained in green. And you see this nice synapse photoreceptor with the bipolar cells. They also contain outer limiting membrane. So in a sense, organoids, these retinal organoids, recapitulate many of the features of the human photoreceptors. However, one caveat is that the nascent outer segments, which are not as mature or organized as those of in vivo photoreceptors. Now, let's look into how to generate retinal organoids. It all starts with stem cells or induced pluripotent stem cells. First step is the induction of the retinal embryoid body formation. Here is an image of the embryoid body. Mostly it is achieved by the self-aggregation property of the iPSA. By the way, please note that all these pictures are not to scale. By day 17, you start to see optic vesicles with stratified neuroepithelium. There are quite some factors and conditions that drive this directed differentiation from embryoid body to optic vesicles. Initially, matrigel was used for this step, but later on, more chemically defined components such as BMP4, bone morphogenetic protein 4, became popular. By day 30, you will have an early retinal organoid with retinal precursor cells. This transition is commonly achieved by Wnt signaling pathway activation and inhibiting the FGF signaling pathway. After this, they are switched to maintenance media where they develop and mature. The first 1 month is the most tricky and difficult part with multiple key steps, whereas thereafter it becomes a bit simpler and more like a routine cell culture. Retinal organoids do take up to 150 days to differentiate. However, there are reports that they continue to mature till 200 days. Once they're fully mature, they are ready for investigations. They should be good for a month or two. People have maintained them for much longer periods, but there is limited data about the quality of these organoids when maintained for very long durations. And retinal organoids can be of wild type, volunteer-derived, or mutant, derived from patient iPSC. Next, I would like to give you a brief flavor to the published protocols. The first landmark studies describing the human retinal organoids were published around the year 2012. Nakano et al. From the group of Sasai from Japan published the landmark paper in 2012. This has been a very popular protocol in the field. Around the same time, Jason Mayer from USA and the group of Melinda Loco from Newcastle, UK, published similar protocols. In 2015, the group of Sakai published an improved version of their previous protocol by using BMP4 for directed differentiation, which has greatly improved the retinal organoid yield. In 2018, groups from Newcastle, UK adapted these protocols to 96-well format, greatly improving the throughput. In parallel, Zhang et al. From the group of Valeria Contosoler published a protocol in 2014, which has also become quite popular. Later, Gonzalez et al. From the group of Robin Ali published an improved protocol to this one. As a disclaimer, by no means this is a complete summary of the landscape out there. If you are new to the field, these protocols may be a good starting point. And there is also a nice Mathers paper from the group of Jason Mayer, which I would highly recommend as a starting point if you're interested. So there are different protocols from multiple groups. Some of these protocols are minor variants, whereas some are totally different. All these protocols have their own pros and cons in terms of efficiency of differentiation, quality of organoids, labor intensiveness, automation, etc. So you have to decide the protocol that best suits your purpose. Moving on, applications of retinal organoids for drug development. Broadly speaking, they can be classified into two groups. First, using iPSC-derived photoreceptors as a direct therapy for people with photoreceptor degeneration, that is clinical use. So stem cell replacement therapy. Second, using these models in the development of other therapies, that is research use. Retinal organoids offer great value at various stages of drug development. They're great tools in modeling of the disease and understanding the underlying disease pathophysiology. They help in target discovery and even the target validation process. Identifying the target is the first key step in any drug development process. So they can add quite some value there. Next, they can also be used for screening the drug candidates and estimating their efficacy and safety. And they also offer great promise for translational research and bridging the bench to bedside paradigm. If you're really interested in the applications of organoids, then here are some nice review articles. And later on in this presentation, I will give you a few examples which illustrate how retinal organoids were used for each of these purposes. Now let's have a quick look into how retinal organoids compare to other model systems. Cell models, they are commonly used in research. However, a few caveats are they lack the extracellular matrix, contain only a single cell type, and have no physiological interactions. Whereas organoids contain most of the retinal cell types, which are nicely organized into a complex stratified structure and show complex interactions. Coming to animal models, especially mouse models, some of the IRDs are not captured in mice due to differences in the pathophysiology. We just seen in the poll that people do agree with that view. Large animal models and non-human primates are excellent for research, and they closely mimic the human retina. However, cost is one of the prohibitive factors. Secondly, availability of large animal models with the desired mutation can be a big bottleneck. And there are also some ethical concerns using animal models, unlike retinal organoid model. Next, explant retina, next ex vivo retina explant model. Since this is human tissue, this is the closest we can get to human retina in the lab. However, there are a few limitations to the explants. First, they're viable only for a few days to a couple of weeks at max, whereas organoids can be maintained for months to up to a year. But the biggest difficulty is to get the explant with the desired mutation. For rare mutations, this is almost maybe impossible to achieve. So these are the main advantages of the retinal organoid model. However, there are some disadvantages and limitations too. First, as I have briefly mentioned before, the outer segments are not fully matured in organoids. So far, generating photoreceptors in vitro with fully stacked membrane discs proved to be elusive. Next, the lack of vasculature, and they do not contain monolayer of RPE covering the photoreceptors. So delivery of nutrients and drugs to the photoreceptors is uncontrolled and non-physiological. But some of the caveats can be circumvented using retina on the chip. In these microfluidic devices, organoids can be flattened and placed on top of monolayer of RPE, and the supply of nutrients and drugs can be controlled. So organoids do offer some unique advantages. Sometimes they're complementary to the other models. Sometimes maybe they are the only model that is available. But it's always advised that you should use various models before deriving definite conclusions. Currently, antisense oligonucleotides are a very promising drug modality for inherited retinal disorders. Already, many of the AON drugs are in clinical trials. So I would like to spend a couple of minutes on elaborating the advantages of retinal organoid models for AON therapies, especially the pre-mRNA splice modulating drugs. Alternative splicing of known genes and transcription of novel genes is really abundant in human retina. So for the investigation of splice modulating AONs, it is essential to use a test system which mimics the human retinal splicing machinery. For example, mouse and zebrafish splicing machinery may not recognize human mutations, so may not be suitable for screening human drugs. I will come back to this in a moment. Moreover, mouse sequences are often different to human sequences. These differences do not need to be very big, and even the SNPs make it difficult to study the actual human AONs in mice. As a result, surrogate AONs may be needed for studies in mice. Also, there is a lack of suitable IRD mouse models with the desired mutation. Coming to the cells, retina or photoreceptor-specific genes such as USH2A, ABCA4 are not expressed in non-photoreceptor cells such as fibroblasts. Also, AON uptake into the cells and delivery to the productive compartment are very slow processes, requiring studies of long duration. Unlike retina, both primary and cancer cell lines divide rather fast, diluting the AON that is taken up. Importantly, naked oligos are not readily taken up by the cells and often require the aid of a transfection reagent. So this is not representative of the in vivo uptake. Hereafter, in my presentation, I will walk you through several examples that exemplify the potential of a retinal organoid model for drug development. First example is that animal models may not recapitulate the alternative splicing. Animal models have a lot of advantages. We just saw some of those, but there will also be specific instances such as these where they may not be very useful. For example, mutations in USH2A gene result in Usher syndrome and non-syndromic retinitis pigmentosa. One such mutation is this C7595-2144A2G. This is an intronic mutation in intron 40. This is also called PE40 mutation. A humanized knock-in zebrafish model was generated for this mutation to help understand the disease and to aid the drug development process. However, this model failed to recapitulate the aberrant splicing mediated by this mutation and thus turned out to be not very useful. Similarly, leber congenital amaurosis 10, LCA10, is the most severe and frequent form of early onset retinal dystrophy. The most frequent genetic cause of LCA10 is a mutation in intron 26, C2991 plus 1655A2G. There are around 2,000 patients in the Western world who suffer from LCA10 due to this mutation. In this case, a humanized knock-in mouse model was generated, again to aid the drug development process. But again, this model failed to recapitulate the aberrant splicing. So for a group of diseases or mutations, retinal organoids may be the only available model. I would like to spend a few minutes on this LCA10 example, because I will show some more data in the next slide. Here is a schematic of disease pathophysiology, and CEP290 pre-mRNA sequence is depicted here. The base G, highlighted in red, is the 2991 mutation. As a result of this deep intronic mutation, a small part of the intron highlighted in this red box is recognized as an exon. When that happens, this new pseudoexon, referred as exon X, gets incorporated in between the canonical exons 26 and 27. However, this pseudoexon contains many stop codons, so the resulting mRNA cannot be translated into full-length CEP290. Interestingly, a small amount of pre-mRNA is spliced into wild-type mRNA. CEP290 protein is critical for the functioning of the connecting cilium of the photoreceptors, which plays a role in the protein trafficking to the outer segment. So absence of CEP290 leads to connecting cilium defect and reduced protein trafficking. This eventually leads to photoreceptor dysfunction and eventually degeneration. Since there are no animal models available for LCA10 due to CEP290, 2991, plus 1655 A2Z mutation, group of Mike Cheatham from London, UK resorted to retinal organoid model to model the disease. Here, they have generated organoids from a healthy volunteer and a subject homozygous for the CEP290 2991 mutation. I will briefly walk you through the data. Let's start with the RNA measures. In control organoids, all the pre-mRNA was spliced into wild type as expected. However, in LCA organoids, most of the mRNA contained the pseudoexon, and level of wild type mRNA was very low. Next, as expected, in control organoids, CEP290 protein is present and co-localizes to pericentrin at the connecting cilium. And in LCA organoids, CEP290 protein was missing. They have also noticed that absence of CEP290 protein results in decrease in the number of ciliated cells and decrease in the length of cilium. Absence of CEP290 protein also results in mislocalization of proteins such as RPGR to the outer segment. They have also made an interesting discovery that showed that the amount of wild-type CEP290 in the LCA organoids decreases as they differentiate into photoreceptors. So there is more wild-type CEP290 at 3 weeks than at 7 weeks or 17 weeks. This explains why this particular mutation causes only retinal phenotype. It appears that the splicing in the photoreceptors is highly prone to this particular mutation. So we in ProCure are working on an AUN therapy for LCA10 due to this mutation. This AUN is called Cure110 and is currently in the clinical trials. As the group of Mike Cheatham modeled the LCA10 in retinal organoids, we jumped onto this opportunity and used them for the investigation of Cure110. Cure110 corrected the missplicing in a dose-dependent manner, in a concentration-dependent manner. Here is a ratiometric analysis of the data where you see the wild-type levels going up and mutant mRNA levels going down with increasing concentrations of Cure110. We did the same analysis with accurate and quantitative ddPCR assays. Here also you could see that Cure110 increased the wild-type CEP290 levels in concentration-dependent manner and decreased the levels of this mutant. We next looked into the effect of Cure110 on ciliation and cilia length. Here are some representative pictures with ciliation markers from various concentrations of QR110. QR110 increased both the percentage of ciliated cells and the length of the cilium. So in the absence of animal models, retinal organoids immensely helped the preclinical development of QR110. Switching gear, here is an example. Here is an example where retinal organoids were used to assess the AAV vector tropism. Gonzalez et al. Tested various AAV serotypes for their efficiency in transducing human iPSC-derived photoreceptors in vitro. Here are some representative pictures. The nuclei are in blue. Red is recoverin photoreceptor marker, and green is GFP signal coming from the AAV vector. All the serotypes were tested were capable of transducing the photoreceptors in vitro. However, AAV serotypes SHH10 and AAV2/2 seems to be the most efficient vectors at transducing photoreceptors. As I have just said, ideally you should use multiple model systems. So in the study, indeed they did use mice, and the results from both these models nicely complement each other. Next, a brief example where retinal organoids were used to identify the underlying cause of the disease. Many of the Stargardt disease subjects harbored this ABCA4 mutation, C5461-10T2C. This is in fact the third most frequent ABCA4 variant in these subjects. However, it was not clear whether this mutation is the cause of the disease because the splicing prediction programs indicated only a small effect on splicing efficiency. ABCA4 mRNA splicing analysis in mutant ABCA4 organoids or photoreceptor progenitor cells revealed that the mutation has a clear impact on splicing. In controls, there is only wild-type mRNA, whereas in patient P1, who was homozygous for the mutation, has no wild-type ABCA4 mRNA at all, only misspliced mRNA. And patient P7 contains a totally different mutation, so of course no effect on splicing. And patient P16 is heterozygous for the mutation, and that's why both wild-type and mutant mRNA are present. So identification of this C5461-10T2C as the causative mutation, that is as the target, allows both to study the underlying disease pathophysiology and to develop drugs that correct this phenotype. So as a final slide, some practical considerations. Many protocols are described for retinal organoid generation. All of them contain a lot of nuances. It is still more of an art than science. Availability of iPSC with the desired mutation can be a bottleneck. However, with the advent of CRISPR genetic engineering, mutant iPSC can be generated rather than procuring from the subjects. And cell line specific variables do or may influence differentiation and even the outcomes you are interested in. And retinal organoids take 5 to 6 months to mature, and the yields depend on iPSC and the underlying mutation, and all batches may not be successful. So long-term planning, staggered production, and arrangements for weekends and holidays are needed. And last but not the least, reagents from cells and mouse may not work for human organoid models. So you may have to set up and test the assays in parallel in advance. With that, I'm happy to take questions.
Thank you. Sorry about that. I couldn't find the mute button. Thank you for that presentation. So how about we go ahead and we start with the polling question this time, and then there's a couple questions that we'll do after that for you, Dr. Dula. So Liali, could you load the polling question, please? Again, this is for the audience to answer. And this is, have you worked with organoids before? So just to give us just a sense, and we'll let this go for about 30 seconds or so until you answer. At the same time, Dr. Dula, let me go ahead and throw a question your way. I see a question came through from the audience, and it is what are the differences at genetic and epigenetic levels, and how will they may affect both organoids and photoreceptor differentiation deriving from different patients' iPSCs?
It's an excellent question. The emerging data indicates the differences are reflective of the underlying genotype from the donor itself. So when you do see differences in terms of differentiation or other variations you see, maybe they are actually reflective of the donors, not so much as the variation between different clones of the iPSC from the donor or cell line to cell line variation.
Okay, and we have one more question here. Is how well does the efficacy in organoids predict efficacy in vivo or in humans?
I think that's again an excellent question, maybe even $1 million question. I think we need to wait for the outcome of some of these clinical trials so that we can correlate the data from the organoids and the clinical results to really answer this question. So until this, it's very difficult to say how predictive they are. But I think they can be quite predictive, but we really need to wait and see the data.
Thank you. One last question actually from me. This is sort of based off of the results from the poll that we just took. And there were a number of individuals who basically said that they have not worked with organoids before. And I'm assuming that has to include retinal organoids. So therefore, if someone is looking to get or sort of incorporate this tool in their lab, what would be your suggestions on how to get the speed the fastest and incorporate this technology?
So, yeah, I mean, the fastest way is to get started, right? There are protocols out there. They're relatively easier and maybe a short time to maturation. Maybe they are the way to start. But if you're really interested in getting fully matured organoids with good reproducibility from batch to batch and well-characterized models, then it does take time. But you can start with protocols which are quicker and faster, and then you can build your way. And you can also maybe collaborate with groups out there. There are a lot of groups right now working on retinal organoids for various genes and diseases. So maybe that's another avenue.
Okay, and one last question real quick. What is your experience on how has the FDA considered organoid models versus animal models in the IND package?
I think they're quite positive and quite open to the organoid models. As I have shown you, there are instances where we may not have any animal model. Even when there are animal models, they may not be represented to human diseases. So I think they're quite open to the organoid models.
All right, thank you. And Dr. Dula, thank you so much for your presentation. We really appreciate it. Our final presenter is Dr. Gus Aguirre, Professor of Medical Genetics and Ophthalmology at the School of Veterinary Medicine at the University of Pennsylvania. His work focuses on identifying the genetic, Gus, I was reading your.
Oh, should I have?
No, it's okay. It's okay.
No, I will.
No. You're fine. You're fine, sir.
No.
His work has been supported by the National Institutes of Health, the Foundation Fighting Blindness, and Seeing Eye Incorporated. He's the elected fellow of the Association for Research in Vision Ophthalmology, the College of Physicians of Philadelphia, and is a mentor of the Institute of Medicine of the National Academies. His talk is entitled Canine Models for Retinal Translational Studies. The floor is now yours. Please take it away.
Okay. Sorry for jumping the gun.
No problem. You know, we're all working in this wonderful virtual world now.
Okay. Good afternoon, everyone. If you don't hear me well, please raise a hand or yell, and I will, I may have a problem with my microphone. So our work deals with canine models of retinal disease, initially to identify the models and then study without ever having any hope of doing any treatment, identifying the cellular mechanisms of disease. About 3 years ago, we set up at Penn a division of experimental retinal therapy, so that we are by definition experts in the field, since that's the acronym that William Beltran selected for this. And it includes individuals within our group at the veterinary school, and then affiliated faculty within the School of Medicine, the Shea Institute, and in this case, the School of Dental Medicine. And we have then other individuals involved with vector development, for example, Bill Housewood, Al Lewin, with whom we collaborate extensively. The traditional pathway from the bright idea to the application to a patient usually began with the idea and then it jumped to a mouse. And usually there was no dog in the way. So you went idea, mouse, person. And then beginning in early 2000s, there was a slight paradigm shift where you had idea. A dog happened to have been a perfect model for the human disease, and then began this translational pathway to humans. And some cases, the mouse was also ancillary to this. And in other cases, the mouse, because they did not have a phenotype that compared to the human was not relevant to that translational path. So in 2001, when we published the first study on RPE65 gene therapy, about 2-3 years later is when the work was done with the mice. Now this is now approved as Luxturna. After many different groups began this work, 3 clinical trials set up in 2 in the US, 1 in UK, and then groups in France as well. In 2007, we showed the proof of concept that achromatopsia, that's a selected disorder of the cyclic GNP-gated channels in cones, was amenable to gene therapy. And, and then that led to what is now a phase 1/2 clinical trial, not only in the US, but in Europe with other groups. Subsequently, with X-linked RP, that's the RPGR gene, we showed that you could treat the disease, either prevent the disease from developing or arrest its progression in 2012. And later on, we showed that you could treat at a very, very advanced stage of disease. That too is in phase 1/2 clinical trials by different groups. Ongoing studies involved with rhodopsin ADRP, where we have a gain of function mutation in this gene, and we need a combined knockdown replacement strategy that has been very effective, and also with BEST1 disease, best vitelliform macular dystrophy. And that is now ongoing with through Iberic Bio. And lastly, we're just beginning the preclinical studies now with LCA, a form of LCA caused by NPHP5 mutations. And this has shown really dramatic, not only recovery of function that was not there, but preservation of retinal structure. So what we do is we identify and characterize naturally occurring canine models. These are dogs we find in our clinic population, or which people bring specifically for second opinion. For many years and still now, we're really interested in knowing the cellular and molecular mechanisms of the disease, because knowing these allow you to develop specific therapies. Knowing this information allows to do therapeutic strategies. Years ago, neuroprotection and gene therapies, and now we're moving towards cell therapy, photoreceptor cell transplantation to reform the photoreceptor layer. And for the past 3 years, we've been able to do IND-enabling studies that have been requested or required by the FDA. So we have many, many models. There are over 20-some models. All of these here have been identified in my lab. We have LCA forms, the RP65 and the NPHP5. We have a number of early onset autosomal recessive RP disorders, like this RD3 mutation could just as easily fit in the LCA category because that's where it fits in human patients. The rhodopsin autosomal dominant RP, 2 forms of X-linked RP that we have, late onset autosomal recessive disease, Conrod dystrophies, achromatopsia, 2 different ones, a genomic deletion and a missense mutation, and then autosomal recessive dystrophinopathy. Most of the dogs do not look like this. They have been mongrelized, they're smaller in size, and that allows us to manage them better. Having a 150-pound dog in your kennel eats a lot of food, produces a lot of poop, and you have to do a lot of cleaning, so that these now weigh about 25 pounds. Just to give you an idea, this is the webpage from our division at Penn. And these are references that go back to, I believe, 2001. And this is just a selection of the ones in 2020. There are studies dealing, in this case, with human patients and with dogs. Here's looking at the axon guidance receptor and how it is involved with retraction of synaptic terminals in a disease. Changes in the postnatal development of the retina. And that's really important because the retina, as a dog matures, thins normally. So when you do a treatment, you want to know the assessment is not at the time you did the treatment, but at the time when you are actually evaluating the disease. What will be the normal thickness? 2 FDA sponsored trials with AGTC, and then another one in review on human clinical trials. So we do this work in 2 places. One is in our lab in the city of Philadelphia at the School of Veterinary Medicine. And then in the rural campus of the university, we have what is called the Retinal Disease Studies Facility, which is a facility built in 1989, combination of funding from FFP and NIH, modernized in 2005 with FFP and slight NIH funding, and modernized again a few years later, where we have facilities for doing clinical exams, OCT with either a spectralis or a bioptogen units, a surgical suite. Now that has been, this is outdated since now we have a Zeiss operating microscope with surgical OCT capabilities, ERG facilities, and these are all modified to fit the dog. The standard downslope domes that are available for people are too small for a dog. So basically our machine shop has taken 2 big turnpike light reflectors and then fuse them together and now put in all the stimulation inside that gunstone unit. Vision testing, either with an obstacle course or a Y maze, and then in the city where we can do the histopathology, immunohistochemistry, and molecular studies. Just to give you an example of some of these studies, some successful, some not. Interesting that Regeneron was funding part of this webinar because in 1998, I think, Regeneron had a product called Axokine, which was a CNTF mutant that they were planning on injecting in people every 3 to 4 weeks based on results in rats. They were very, very effective. And we did this work in dogs that we would inject at this time point, repeat at this time point, and then at about 14 weeks, we would go ahead and assess the outcome. And Axokine was extremely effective in preventing further photoreceptor degeneration, as you see here, the little triangle, except that the treatment was worse than the disease. It caused cataracts, uveitis, recurrent epithelial erosions. That was modified subsequently by a company called Neurotech, where you had a slow release encapsulated cell therapy device, which is implanted about this age and then assessed at about 12 weeks later. And again, there's excellent preservation with minimal to no adverse effects. What it is really seen here is whenever you do these studies, the affected dogs have a very characteristic and predictable rate of degeneration, and the normal dogs. So we can do this over and over again, and the results basically for control and affected dogs are the same. The CNTF work was carried out by Paul Seeding at NIH for both early and late RP and for geographic macular degeneration. And unfortunately, those results were not effective. And the use was discontinued for that. There are other drugs that we have used in cooperating with pharma or investigators or ourselves, and that have not been effective. Diltiazem was proposed in the 90s by a group, late 90s, by a group who did this work in mice. Amazingly, it was published, I think, in Nature Medicine. Should never have been published in Nature Medicine or anywhere. But we did this because of the FFB's interest as a potential treatment as a calcium channel blocker that we administered here. The green shows that there's absolutely no effect. And subsequently, studies in rats and mice by other groups showed that also it's not effective. We have used a neurotrophin vector that's proprietary to one of the companies that we helped through FFB, and it caused no benefit. Another one was almost a stem cell therapy that was cells implanted into the vitreous or the subretinal space to act as a neurotrophin pump. That was not effective. And that's one of the reasons that they did not go further in the development of this product. It was only effective if the cells caused a retinal detachment. The retinal detachment slows degeneration, but obviously that's not a therapeutic alternative. Let me give you a couple of examples of studies that we've done with X-linked RP caused mutations in RPGR. In 2012, as I said, we showed that we was effective in either arresting the disease or preventing it. A few years later, we then showed that if you delay the treatment, not to when it's early, but in the mid or late stages of the disease, at a time when you've lost 50 to 60% of the photoreceptor, treatment in these areas you see the dashed lines arrest further progression of the disease. You see that here. This is a lower dose and it was not as effective here. The ERG is, again, 3 years roughly after treatment. You've had preservation of rod and cone function in the treated eye and absent function in the untreated eye. And then that allows us to work, in this case with AGTC, with a codon optimized vector that was here for RPGR, and that it showed that the codon optimized was comparable to the stabilized RPGR transgene that we have been using. Best macular dystrophy is another example. There's comments made about the presence or absence of macula in animals that are not primates. Dogs do have a foveomacular area, and this is an area with a high cone density, and that is the area that is affected by inherited macular degeneration, either Best disease or a RPGR, where you have a macular disorder early on in a type of mutations. And in this disease, you have the area with a large vitelliform lesion with a pseudohypopium here in the dog, and the human, the areas that you have lesions in the foveomacular region in people and dog with satellite lesions, as you see here, or the autosomal recessive dystrophinopathy, where you have multiple lesions throughout fundus and a quite aggressive and severe disease. Dystrophin is a channel, a calcium-mediated channel that is located in the basolateral RPE cell layer. This is in a normal, and the disease is amenable to treatment. So we first developed vectors testing in the normal dogs about 6-7 years ago, identified vectors that were effective and also vectors that cause damage to the retina, and then went on to treat dogs with this disease. Here's one, a dog with a large pseudohypopium treated. Here's the area where you insert the cannula in the subretinal space, a little focal scar. The lesion disappears. And now you see this this picture was taken a year ago. I don't have the latest pictures that were taken September of this year. So it's 8 years, 4 months and 17 days since the treatment, and the lesion has remained gone. And that we published recently in PNAS, study with Art Sadesian and that was led by Karina Gushevich. So who are we and what can we do to help you? First of all, we're university-based. We're not a CRO. All of us have faculty appointments at the university. We're supported by specific facility grants from the FFP and NIH to, and the emphasis here is assist in proof of concept studies. At one point, NIH funding was substantial. In the past 8 years, it has been reduced for 45%. Their idea was that there's no inflation, so cost of living doesn't go up. And if they give you less money, you do the same amount of work. What we could do for nothing many years ago, we cannot do now. So it's neither FFP nor NIH supports helping you to do the work. This has to be provided by the PI or by the company. This is made through the FFP. We're also not able to do your research. If you decide, oh, I'd like to do 10 different things on this, this is a great project, wonderful. We're happy to do it, but not through this agreement. This would have to be done separately through the University of Pennsylvania. And obviously there, the cost goes up. So best way to do it is we have discussions with you. We select a model as suitable for the question you want to ask, determine the route of therapy, set up a protocol agreement, and we then present the budget to FFP. FFP discusses the budget with the PI. And at that time, we have to then write an IACOC. We will not do anything until this budget is approved by both the PI and the FFP. And the reason is that we've put a lot of work on this at times, and people decide, oh, I'm not going to do it. The IACOC application takes 2 to 3 months, sometimes a few weeks less, often a few weeks or months more. Dogs don't come from the shelf. We have to breed them. They breed twice a year. They have a gestation of 63 days. And if you do a study at 6 to 10 weeks of age, you take You have to have 63 days times 70 days before, if it's 10 weeks of age, before you can do the study. And you have to plan that the dog is going to be bred. So we do not like what I call only child in this program. If you are an only child and you expect to get what you want immediately, it's not going to happen, because if there's a queue of people who would like the dogs, would like to do the studies. So we have to prioritize this based on when you came in with a question and agreed to do the study, and when dogs become available for you. So is what I put here: if you desire 10 dogs at 6 weeks of age next month, forget it. It's not going to happen. Only if you're extremely, extremely lucky that someone who did want the dogs and was in queue to receive them, their product did not work, and they decided to cancel the study. As examples of what we're doing now is, again, I mentioned the rhodopsin ADRP and Best disease. They are scheduled to go into treating patients in a phase 1 trial in 2021, hopefully about middle towards the end. The X-linked RP is being continued separately from AGTC with a vector and a transgene that is proprietary to the university. We have the MPHP5 LCA that is now in pre-IND enabling path. All the work has been done on this. Stationary night blindness caused by mutations in the LRED3 gene that is present in bipolar cell dendrites. We've had now excellent results with gene therapy, either administered intravitreally, which is not as effective, but putting it subretinally and targeting the on bipolar cells. Extremely effective. We have a new form of LCA causing a foveal macular degeneration. I'm not putting the gene here because that still paper is being written. We have a model of ABCA4 Stargardt macular degeneration that has now just been has been identified about 2.5 years ago. And we now have expanding the breeding colony for studies. And we have also a new phototransduction gene defect. Again, paper is being written on this and hopefully will come out. It's too late in the year, not by the end of the year, but early next year. I think that's the last slide. So I'm happy to answer any questions.
Thank you, Gus, for that wonderful presentation. Really appreciate it. I guess we'll start with just a couple of questions, and I had one for Dr. Gate. It's could you maybe elaborate a little bit on how the particular, you showed that nice sort of slide that showed the different dog models that you currently had. And I was wondering about how were those identified, and are more dogs sort of in the list to be developed or added to that list that you showed?
Okay, there are the three that I included at the end are not on the list yet.
Okay.
I was lazy and I didn't want to go looking for a picture of a dog. There are a few others. We have an S antigen Gucci's type disease that we have, but it's a very slow disease process. It's not really worthwhile at this point using it as a therapy model. Many of the dogs many of the models that are not of great interest to other investigators or to us, we keep frozen as frozen semen, so that we will then go ahead and take semen, breed to a normal or a carrier female, and then do back process and be able to produce a progeny. That takes longer. So we sort of tailor the breeding animals available to what the expected needs are. So for example, at one point there was a great need for the RP65 mutant. We used them, we did several papers on it. Then the interest died because there was now Luxturna or on the way to Luxturna. And then when we and others found that perhaps Luxturna is not, the cure is not permanent, that the cure may be effective for a few years and then it decreases, then there's a revived interest. And we have been using those, and we maintain a small colony ready to be developed if someone else shows an interest.
Okay. And also during the testing part, what other sort of bioassays are you looking at? Are you collecting urine? Are you doing blood draws and that sort of thing as well?
For all the gene therapies, we collect pre and post samples of blood. We do those and we free serum for this for the future. We have not been collecting urine. That is, again, it will be at the request of the investigator. If urine is requested, we will collect urine. That's usually a little bit more difficult in dogs because it usually involves a bladder top, which is very, it's not, it is done routinely in the clinical setting, but you're not able to do it on an hourly basis. You do it infrequently. So urine collection is at tiers. We collected at one point for looking at vector distribution and see whether it was excreted in the tears. We have several pathologists who do the GLP type pathology work as reported and accepted by the FDA.
All right, thank you. I believe we have one last polling question for the audience. Leala, could you load that, please? Here. And this question is, in what capacity have you worked with a large animal? And while we're waiting for everyone to respond to that poll, we'll leave it open for about another 20 seconds. I'd like to again thank our panelists for wonderful presentations. And as soon as this poll is over, we're open for a general discussion. I will remind you, if you have a question, please enter it into the Q&A box at the bottom of the Zoom application. So yeah, so we can get the Q&A part started. So here, just from that poll, it seems that it's about even that most people, like most have not worked with a large animal model. Some have only worked it in the setting of a farm and tox study. And the last one is the work with a large animal because of naturally occurring mutation or disease. So it seems that a nice sort of spread of representation there.
Well, Chad, that's one of the purposes that FFB and NIH set up this program, because most labs are not set up to have the animals. Most labs are not supposed to have the personnel to handle them. So that's why we're able to do it in a quite cost-effective basis for the investigator.
Very true. Like I said, we'll open it now for the general discussion. So the other panelists, as well as my co-host from Odelia, please unmute your lines and let's start the discussion. We have about 15 minutes left for the webinar section. So please feel free to jump in with a topic or a comment or question, and we'll begin.
Chad, I have a question actually for Gus. Are you guys normally running just a focused efficacy study, or do you try to combine efficacy and pharmacology toxicity studies in the dog studies you're running?
Depends. If it's studies for us, yes. If it's studies for others, we tend to do first a proof of concept. Does it work? And then if that works and they're interested in doing it, then we go for the next level. For example, everybody says, we want to incorporate OCTs and ERGs in everything we do. We want to use the RCD1 model. RCD1 model has and they're not using a gene-specific therapy. They're using something else, let's say a neuroprotective strategy. Well, rods don't function when you have a beta PDE deficiency. And the cones function for a year and a half. So in a short-term study, an ERG is irrelevant. OCT, again, in a short-term study is irrelevant because the gold standard is pathology. if the short-term study works and is really effective, and the PI or the company wants to do a long-term study, then OCTs are critical, because why plan a one-year study and then wait until the end and find out that nothing worked? So if you incorporate OCTs into the treatment at 3 or 4 month intervals, then you know ahead of time if the product is working, and have a no-go or go decision at that point. So it depends on the study, but everybody in pharma, there's been, and especially CROs, when they deal with companies, everything is OCT. Do an OCT, and I guess it's a billable procedure. And a billable procedure makes money. So it's, you sell them, but here, when you're limited with personnel and space, doing a procedure that is really irrelevant for what you really want to know, which is the treatment effective? Did it prevent degeneration? Did it preserve the photoreceptors? Histopathology is a way to go.
Great, thanks.
We have a question from the audience for Gus. It is there a possibility to develop a canine model for ushers?
Interesting. I think it's, I would not hold my hopes very high. We have identified, editorially, a group at Penn identified a mutation in one of the usher genes, and the retina was normal. And so far, really, most of the usher genes, except in people, do not cause a retinal phenotype, or the retinal phenotype is quite mild. If people with ushers, I think it's 1B, which is myosin 7A, if they had the same phenotype as a mouse, they wouldn't worry about blindness. They wouldn't be blind, because what you're having is moving of a lack of movement on microtubular network of the melanosomes in the RPE. But the photoreceptors don't degenerate. Or if they degenerated at a time that a person would have been dead. So it's a real problem. I think it really would be, I think non-human primates would be the way to go for looking at usher models. I think they're the ones likely to show something that is really significant. And it may be, and the question to me of interest is, which no one would ever fund me, so I'm not going to ask, is why does a human having most of these Usher syndrome mutations such a severe retinal phenotype, yet the mice do not? And that's a basic biology question, but everybody wants to do treatment, and no one wants to do basic biology.
All right, thank you. I think that answers the audience question.
I have a question for
yes.
Caroline, I have a question for you. Can you comment on organoids between human and mouse, and similarities, differences, values of using something like a mouse, and if humans are organoids aren't, somebody's not as well versed on that, or what would be the advantage, disadvantage?
So the differences between mouse and human organoids, right? There are quite some advantages to mouse organoids because they do differentiate rather fast. So you don't need to wait for all 5 to 6 months. I think the protocols are like around 2 months. So they're really quick. And so that gives you a much faster way of looking into the disease biology and pathophysiology. And not all diseases are so different between mice and humans, right? There are some diseases which nicely recapitulated in mice. For those, I think organoids, mice organoids really are a good tool. And someone asked the question, how well they predict organoids in vivo response. So in humans, we cannot do these experiments other than clinical trials, but we can do these experiments in mice. So that way you can do mouse organoid studies in vitro and do the same study in vivo and also sort of correlate and build the sort of technology. So that's a kind of a platform application.
Thank you.
All right, Dr. Dula, I have a sort of follow-up question to that, and I may sort of stumble through it, so bear with me. Is it known what are sort of the physiological or, let's say, outcome differences of retinal organoids generated from different patients with the same mutation? And I guess what I'm trying to get at is, for a number of years, I worked in cancer biology. And if you think about drug treatment, somebody may have a similar type of cancer or the same mutation that's driving the cancer, but when they take the drug, they have different outcomes due to their personal physiology. So I'm trying to better understand from a personalized medicine aspect, are there slight differences between patients with the same mutation?
I think, to my understanding, these studies are not done really thorough studies where you take the same mutation and generate the iPSCs the same way and you differentiate the organoids the same way. These are really time-consuming studies. So most of the data we have comes from different labs using different protocols and different reagents. So it's still to be addressed, but the emerging data indicates they do, to a large extent, faithfully represent the donor characteristics and phenotype, but they still need to be confirmed by fully controlled studies. And also, you need to keep in mind this technology is not there for long, right? The first studies were described, first reports were made in 2012, so it's only like eight years. So I think in the coming few years, I think we see more and more of these kinds of data that will help us really take it to the next level and use in precision medicine.
Sorry, Ashley, did you have a question?
No.
Okay.
Actually, well, but now that you say that, I was considering with the organoids seem to serve a very specific place. So I'll continue off of that thought, because I was thinking during your presentation a little bit about what the regulatory path then looks like off the back end of organoids. They're a very specific place when you talk about splicing factors and trying to model human tissue rather than moving through mice or other species. What does the safety path look like right before you move into clinical trials? How are you thinking about farm tox?
I think the toxicology, the gold standard is GLP tox studies in non-human primates or rabbits or some instances rodent models. I think there still are the sort of standard FDA needs for approving any IND application. The organoids can be a great tool for safety screening, for instance, or kind of weed out some of the bad actors before they enter the expensive NHP tox studies, GLP tox studies. But I'm not aware that they're replacing the formal GLP tox studies at this point in time.
Thank you.
Dr. Hayter, I had a quick question sort of about your views on sort of the future of gene therapy. I think that with Luxturna being out there and its great success that it has sort of busted open the door for, a real push for gene therapies. But looking at inherited retinal diseases, and it's such complex and so much diversity genetically amongst these diseases, it doesn't seem feasible to create a gene therapy for all of them. So what is your sort of view on the future of that? How do we get, to more therapies for this, whether it be either gene therapy or something else, or a pan disease therapy? So I guess just your futuristic view, what do you see in the next 10, 20 years for how to address these things when there are sort of viable things available, but just the money and time that, really we don't have?
Yeah, I mean, I think you hit on the major points that it's not feasible to make 200 individual therapies or however many they end up being. And that's just considering your monogenic retinitis pigmentosa type diseases. Then if you look at the retina and the more common complex diseases like AMD, you're taking that to another level of how do you make, and then a broad spectrum therapy. And that's kind of where our lab has been sitting for the last decade or two, thinking, taking the step back and saying, okay, all of them have a common problem. They may arrive at it in different ways, but when we did our transcriptional studies and kind of bioinformatics, it's networks and pathways that get impacted, whether it's a mutation in rhodopsin or a mutation in PD6 beta or ABCA4, et cetera. And then when you have a combination of those. So I think the future is going to see more of these broad spectrum type therapeutics come about. And I also think one other thing that's going to happen is combination therapy. We didn't talk about that much, but I think when we think about viable, feasible to address issues of, sensitivity to dosage, whether you're taking a master regulator, what else is it going to impact, and how can we really create a retina that's in a healthier state, and that healthier state will support the cells that don't even have the mutation but are dying now, right? So I think combination therapeutics that are geared towards something that address like oxidative stress, increased metabolism, et cetera, those will come into play more. And that has, and that's the level of, what's your target. Then I think the other two exciting parts are going to be what other vehicle of delivery, and can we get sustained effect? Can we do this with, even if you have to do an injection, can you do it where you can do a one dose, and it'll actually have a longer term effect rather than having repeat doses? And then looking at things like we talked about earlier with the drops versus contact lens or some other delivery that isn't as invasive as an injection. But I would think we're going to see a lot. I think if we've learned anything from the Genome Project coming out, is every year it's really cool and exciting to see all the different technologies coming through. And it's a fast-paced field, and the door was busted open now. So it's going to be a lot of different technologies coming and trying. And that's why these models are so important, whether you're using organoids or using large or small animals, to really test out what would work well for humans.
Yeah, and to follow up, Nina, I completely agree with you. I think looking forward, with precision medicine approaches, it's just becoming more and more doable and preferable to really try to tackle genetic therapies and get at the root cause. But Chad, I think your point, it's challenging. The price point, the manufacturing around these, it makes that's a hard bar for every rare disease, because a lot of these sit in the rare disease space. But I think once you start to look at new delivery technologies, if we start to put pressure on the manufacturing process, when costs start to fall, if you can apply these, even if it's just to your point where we're starting to see that durability wane, if you can reapply it through a drop rather than such an injection process, you really start to then change the landscape on these things. And maybe they don't go away. Maybe gene therapy just becomes so much more doable. But I think it's going to take pressure points on the system in different ways. And the models become so important to that process.
Yeah, I agree. I mean, I could even see a combination therapy where you end up, even if you start with an injection, you follow up with drops, right? Just to maintain that durability, that sustainability of effect.
Okay, yeah. So I see that we're at time. However, Gus, I'm going to give you the final word before we close the session.
When you look at these diseases that we treat, these patients have lost about 25, but usually about 50, 60, 70% of the photoreceptors that are being treated. And it's really surprising that the remaining cells can tolerate a subretinal injection with a burst of fluid coming in and not die. It's like you have a tsunami coming in and you're trying to hold back the water. And surprisingly, they're resilient cells. So having ways you can treat that minimize that damage to the subretinal space, whether it's intravitreal, suprachoroidal ways of doing it. For example, the encapsulated cell technology or neurotech was a wonderful way of delivering to the eye proteins that they were in a membrane that was not attacked by the immune system. That was just super. Sadly, it did not move forward the way I had hoped it would do. So that the failure with CNTF encapsulated cell therapy devices, I had hoped that it would have translated to other products that can be used for that type of delivery.
All right, and with that, I would like to, again, thank all of our panelists for your wonderful presentations and your insight. I really hope that the audience enjoyed the presentations. Lastly, I'm hopefully sharing with the correct screen as everyone's seeing, so thank you for your attendance. Our next webinar will be in January 2021, which will be working with contract research organizations and contract manufacturing organizations. If you have any questions or comments that you'd like to follow up with us about that, they're available at these email addresses. And again, this presentation has been recorded and will be available for viewing at a later time on the Foundation's YouTube page. With that, we'll close the session. And again, a big thank you to our panelists today. Really appreciate it. Everyone have a wonderful, wonderful rest of the day, and we'll talk to you later.
Thank you.